Well, good morning. It's really my pleasure and honor to introduce today's Montagna Lecture. I thought I would say a few words about William Montagna first, though. I think many of the younger people in the room probably have only a vague idea of who he was. William Montagna was a giant in cutaneous biology. He was actually Italian, born in Italy in 1913, came to the United States as a young boy, and became an American citizen in 1927. He was not a dermatologist. He was a PhD scientist. He began his academic career in 1944 as an instructor at Cornell University, which is actually where I am, so I'm delighted by that. He is best known, I think, for something that he did when he had moved to Brown University, and that was that in 1950 he started a series of seminars on the biology of the skin, which were originally called just that, biology of the skin. He later moved to Oregon, where he became the director of the Regional Primate Center and continued those. He sponsored these various symposia until 1980, and we now know these as the Montagna Symposia on the Biology of the Skin. And I'm sure many people in this room have gone to some of these. I urge you to go. They are terrific, really terrific scientific meetings. Just to point out, in the past, people who are active in these meetings include some of the very well-known giants of our past, such as Tom Fitzpatrick, Aaron Lerner, Walter Lobitz, Irv Blank, and of course we have the Irv Blank Forum at this meeting, Harvey Blank, et cetera, et cetera. And so these really are terrific meetings. So Al Kligman referred to Montagna as the, quote, greatest anatomist of the skin of the last half of the 20th century. He had written over 400, or has written over 450 scientific articles, 6 books, a very productive, very busy scientist. On a personal level, it was said he only needed 4 hours of sleep and would be in the laboratory all day and all night. He also was someone who liked to have a good time and had squeezed pleasure out of many aspects of life. He is now honored here, of course. In 1975, the annual William Montagna Lecture was initiated. This lecture is intended to honor and reward young active investigators. And today we have an absolutely terrific young active investigator, Michael Rosenblum, who's going to speak to us. Michael is a native of Canada. He attended the University of Western Ontario, received his MD and PhD degrees at the Medical College of Wisconsin, then did his clinical training at UCSF, did research training there, including a stint with Abul Abbas as a research fellow. He is on the faculty, has risen through the ranks, is now an outstanding immunologist who's made many contributions to the understanding of immunobiology, particularly immunobiology of the skin. His work is focused especially on regulatory T cells, which he's going to talk about today. I am absolutely delighted that he is going to speak with us. And now, Michael.
Thank you, Rich, for that really awesome introduction, the emphasis on young. I really liked that, was the best part of it. But it's really an honor and a privilege to be here. I'm really excited to talk today and address my colleagues in this format. So thank you to the organizing committee for giving me this opportunity. So I do have relevant disclosures for this talk today. I am a consultant for Celgene, and I'm a co-founder and consultant for a biotech startup called T-Rex Bio. Okay, So, this is the problem. As many of you know, as practicing dermatologists, the skin is no stranger to a multitude of autoimmune and chronic inflammatory diseases. And I would argue that the skin probably is the organ that's most affected or has the most autoimmune and chronically inflammatory diseases that affect it. And really, the way we used to think about this disease is that individuals that get these diseases have autoreactive lymphocytes that escape central tolerance, either in the bone marrow and the thymus. They escape into the periphery, and they then see their antigens in peripheral tissues and are capable and actually mediate inflammation. And for a long time, that's how we thought about the pathogenesis of these diseases. But we now understand that everybody has these cells. So everybody has, and as Yasmin pointed out, this pointer's not working, I'll do my best, but everyone has these autoreactive lymphocytes that actually escape central tolerance. So the question then becomes, why do very few of us go on to develop these conditions of the skin? And thankfully, in the general population, they're still quite rare. And the answer now that we know has been sort of a paradigm shift in immunology and the way we think about the way the immune system works. And that is because we now know that these cells, these autoreactive lymphocytes, are constantly being regulated. And they're constantly regulated by populations of cells called regulatory cells. And we know now that patients that go on to develop chronic autoimmune and inflammatory diseases of the skin actually have a disruption in this balance. And that really understanding how this balance is established and maintained will allow us to better understand the pathogenesis of the diseases we see clinically, but also develop new therapeutic strategies to actually treat these diseases, one of which I'm going to talk about today. So, by far and away the most well-studied and I would argue robustly defined regulatory cell populations in both mice and humans is the regulatory T cell, or the Treg. These cells play indispensable roles in suppressing inflammation. Both mice and humans do not have functional Tregs, succumb to autoimmunity quite quickly in life. We now know, thanks to pioneering work by Shimon Sakaguchi and Sasha Rudensky, that there's a lineage-defining transcription factor, FOXP3, that drives the differentiation of these cells. These cells can be derived in the thymus. They also can be derived in peripheral tissues. But most importantly, I think, for this audience, is that these cells really love to live in barrier tissues. So there's a growing body of evidence that suggests that these cells stably reside in barrier tissues, such as the skin and the gut. So there's been a lot of emphasis and a lot of study in these cells and what they're doing in tissues. And they not only have a traditional role that I've outlined in actually suppressing inflammation, but they have these sort of non-traditional or specialized functions in tissues. And this has been basically pioneered by Diane Mathis's group, where she's shown that Tregs that migrate into visceral adipose tissue play a role in glucose metabolism and lipid metabolism. She's also showed that there's a population of Tregs that migrate into muscle and actually mediate muscle repair. And some work done by Sasha Rudensky has shown that even in the lung, there's a population of Tregs that actually have specialized functions in epithelial repair. So one of the fundamental questions of my lab over the last 7 years is to really try to understand the fundamental biology of regulatory T cells in skin. And what I'm going to start today is to talk about a little about how we phenotype these cells and then go on to some functions that we've elucidated over the years. So what is the phenotype of Tregs in skin? We know that there's a lot of them there. So as you can see here, compared to the lymph nodes, mouse skin has 50% of the CD4 compartment is actually regulatory T cells, and this varies with the hair follicle cycle, but on average it's about 50% of the CD4s are actually Tregs at any one time. This is the same is true in human skin, at least the same trend is true in human skin, and that a larger percentage of Tregs are actually found in human skin compared to human blood in the CD4 compartment, and these Tregs have in a memory phenotype, they're all CD45 RO positive. When we transplanted human skin on the backs of immunodeficient mice and asked whether these Tregs migrate out of the skin with time, what we found was that the non-Treg compartment, so the CD4 positive, FOXP3 negative, CD25 negative cells, these T effector cells, readily migrated out of the skin. However, the Tregs stayed put. So we think that in the steady state at least, and we've done parabiosis experiments in mice to confirm this, that in the steady state, these cells are a relatively tissue-resident population. So in work that we did earlier on, we noticed that these cells actually like to be around hair follicles in skin. So they primarily localize around hair follicles and murine skin, and that's shown here. Same is true for human skin. So if you take this is a large hair follicle of human skin. You can see the outer root sheath here is studded with regulatory T cells. And this is the quantification of regulatory T cells in patient biopsy specimens. And what you can see is that in the head and neck region, there's actually more regulatory T cells than the trunk and the extremities. And this is quantification by flow cytometry. And this doesn't correspond with gender, and it doesn't correspond with age, but it actually just corresponds with site. And the correlation here is that these sites have a higher hair follicle density, so we think that the hair follicles are really essentially an important niche for Tregs in this tissue. We've also started to do some single-cell RNA sequencing and some, as I'm going to tell you a little bit more, cytoph analysis of Tregs in human skin. And so we pulled these populations out. No surprise that it's not a homogeneous population. There's heterogeneous population of Tregs in the steady state in murine skin. And we've actually went forward and sort of compared these Tregs to Tregs at the single-cell level isolated from other barrier tissues, such as the gut, and here is shown the lung. And although there's different subsets of Tregs in lungs, when you actually aggregate this data, we can see that actually the skin Tregs and the lung Tregs, they actually, on Tisney plot, actually segregate with respect to the tissue of origin, suggesting that these cells are very unique in the Tregs in the skin versus the Tregs in other barrier tissues. And so this is work that's ongoing in my lab. So as a summary to the sort of the first part of the phenotypic sort of characterization of Tregs in murine and human skin, I think we can say that the skin is home to a large proportion of the body's Tregs. The Tregs in skin have this sort of effector memory phenotype. The majority of Tregs in skin are tissue-resident cells in the steady state. They actually migrate a lot during inflammation, but in the steady state, they're actually relatively tissue-resident. The majority of Tregs in both human and murine skin localize to hair follicles, and that a unique subsets of Tregs reside in human skin, of which we're currently trying to discern the function of the unique subsets. Okay, so function. This is the big question. What do Tregs do in the skin? And to tackle this, my group has really taken a sort of both a basic science and a translational approach. So the basic science approach is we basically use fundamental sort of mouse models, transgenic mouse models, to mechanistically dissect how these cells function in murine skin. And then also at UCSF, we've established what we call this UCSF Cutaneous Immunology Center, where we try to understand the function of lymphocytes in human skin. And we do so through a variety of ways, through ex vivo functional analysis of these cells, as well as humanized mouse models. And so really what our group has really evolved to do is sort of take discovery approaches that we find in human tissue and go back to the mouse to mechanistically dissect pathways, and also pathways that we're interesting in studying in mice and see whether they're actually functionally relevant to the human condition. And so what I'm going to talk about today is some stories out of the basic science program in my lab, as well as the translational program at the end. So dissecting the function of Tregs in skin, mechanistically doing this, we started to think about this question, started thinking, well, what are the unique functions of skin? And maybe Tregs have a role in these, as I've told you, that Tregs are specialized. They have specialized functions in the tissues that reside. So what are the unique functions of Tregs in skin, and maybe Tregs play a role in these processes? And so one unique function of skin is obviously to house hair, and hair follicle cycling is unique to skin. And the other is the skin is constantly, the epidermis is constantly being abraded. And so a unique function of skin is to constantly be repairing the epidermal barrier. And these are two stories I'm going to talk about today. The first is the hair follicle cycling story, which was done by a really talented postdoc in the lab, Nuwa Ali, who now is running his own group at King's College in London. And Nuwa was very interested in trying to understand whether Tregs may play a role in hair follicle cycling. And what sort of initiated these experiments, or this question, was the initial observation that Nuwa made was that Tregs actually like to be around hair follicles, but a subset of Tregs actually really like to be around the bulge region of hair follicles. And as we all know, the bulge region is the home of a very important hair follicle stem cell population in skin. And George Katsourelis and Elaine Fuchs and others have done pioneering work showing that these hair follicle stem cells are intimately involved and actually responsible for hair follicle cycling. So the question then became, do Tregs actually play a role in hair follicle cycling, and possibly did they do this by influencing hair follicle stem cell biology? And so we did what I describe as one of the easiest experiments you can think of, is we just simply depilated or plucked the hair from mice in mice that actually had Tregs, or mice where we could transiently deplete Tregs. And these are the FOXP3 DTR mice generated in Sasha Densky's lab. And then we just actually measured hair regrowth and the length of the hair follicles with time after depilation. And what we found was quite striking, is that mice that actually did not have, mice that actually did not have Tregs, or had Tregs transiently depleted, actually failed to regrow their hair with the same kinetics in which wild-type mice did, or our controls. And what we found is histologically, these hair follicles in the control conditions grew into the subcutaneous fat, showing really nice telogen to anagen transition. These are all anagen follicles. But in the Treg depleted mice, we didn't see this anagen generation, these anagen follicles, suggesting that Tregs were somehow facilitating this telogen to anagen transition, which we know is hair follicle stem cell dependent. So some great work done by Chris Nagao, who used immunofluorescent microscopy to actually look at different markers that define different areas of the hair follicle, sections of the hair follicle. We adapted this approach to a flow cytometric approach. And what we did is we sort of used flow cytometry to look specifically at the stem cells, the hair follicle stem cells in murine skin, and asked how Tregs were potentially affecting this population. And you can see that in the absence of depilation, there's really no hair follicle cycling. This is Ki67 on the y-axis. And when you depilate or pluck the hair from the mice, you can see that a lot of these alpha-6 positive stem cells go into cycle. So they start proliferating. But if you depilate and deplete Tregs, you can see these hair follicle stem cells really don't go into cycle. And that's shown here. It's not just proliferation that Tregs are affecting. They affect hair follicle stem cell differentiation. We pulled these cells out and did RNA sequencing on them and found that in the absence of Tregs, many of the hair follicle stem cell differentiation genes were attenuated, suggesting that these cells actually are involved in hair follicle stem cell proliferation and differentiation during the hair follicle cycle. We also showed not only in a depilation model, but in a natural hair follicle cycling model, the same was true. And so I can't get into the rest of the details and mechanism based on time, but it's summarized here. What NUA found was that a subset of regulatory T cells localized around the bulge region of the follicle. Those cells interact with hair follicle stem cells through a notch signaling pathway is one of the pathways. We've shown that this is actually facilitating their function. They're able to actually induce or help facilitate the proliferation and differentiation of these hair follicle stem cells during the telogen to anagen transition. So we think this may have clinical implications, specifically for alopecia areata, which we all know is a well-defined autoimmune disease. Ralph Paus always gets angry with me describing it this way, but I think that alopecia areata is immune-mediated dysfunction of hair follicle regeneration. And what was interesting is Angela Cristiano's group, she did some great work looking at GWAS hits in alopecia areata and found that, if you're a Treg biologist and you look at this signature, it basically is a Treg signature, CDLA4, IL2, CD25, all genes that are shown to have SNPs in alopecia areata patients. And I think the functional data in humans has been there is a proof of concept in this paper that came out a couple of years ago that used low-dose IL-2 therapy to selectively or preferentially augment Tregs. And I'm going to talk a little bit about this later in my talk. This is a dirty way to do it, but it actually can work. And in a subset of patients, a small cohort of patients that they studied, they were able to augment Tregs and actually induce hair growth. So we think that this biology may be relevant to human hair follicle cycling and human disease. Okay, so what about another function of the skin, which is epidermal barrier repair? And so I'm going to tell you a story about a very talented postdoc in the lab, Anu Mathur, who interrogated the function of of regulatory T cells in epidermal barrier repair. Anu is now since left the lab and is a principal scientist at a biotech startup in the Bay Area. So as I pointed out, hair follicle stem cells, their default pathway is to play a major role in actually hair follicle cycling. But with some great work done by George Katsarelis, his group back in the day showed that actually hair follicle stem cells have an alternative pathway. And in the context of epidermal injury, these cells actually can differentiate and migrate into the interfollicular epidermis and help repair the damaged barrier. And so Anu became interested in trying to understand whether Tregs played a role in this process of hair follicle stem cell biology. And so to do that, he developed a model with the help of Thea Morrow at UCSF of epidermal barrier injury. So this is gentle tape stripping of the skin, and then he measured the recovery phase, so the ability of the epidermis to repair itself after, again, this gentle injury. And he did this in the presence and absence of Tregs to ask whether Tregs play a role in this process. And so what he found was that when Tregs are transiently depleted, there is increased transepidermal water loss through the epidermis, suggesting that Tregs are playing a role in the ability of the epidermis to repair itself after this injury. Looking at keratinocyte differentiation genes known to be involved in barrier repair, Anu found that filaggrin and bleucrin, keratin-1, loricrin were all attenuated in mice that were depleted of Tregs during this recovery phase of the after injury. And interestingly, histologically, what Anu found was that the normal control mice actually have this acanthotic response or hyperproliferative response of the epidermis in response to epidermal injury, but in the Treg depleted mice, this acanthotic response was significantly blunted. So Anu then went on to ask whether this is any way impacting hair follicle stem cell biology, as I mentioned before, these cells play a role in this process of epidermal repair. And so what Anu did is generated mice which he could deplete Tregs and track hair follicle stem cell progeny in a lineage-specific way. And so using these LGR5 TD tomato mice crossed to the FOXP3 DTR background, Anu used his model of barrier disruption where he depleted Tregs, and he actually gave tamoxifen to induce labeling of the hair follicle stem cells and asked how they changed with and without the presence of Tregs. And what he found was at a specific time after injury, these hair follicle stem cell progeny, is shown here in red, readily migrated out of the bulge region and were able to migrate into the interfollicular epidermis and help repair the barrier. But in the absence of Tregs, these cells just stayed put. And this was kind of a striking result. When Tregs were transiently depleted, we basically only saw these hair follicle stem cells retained in the bulge region. There was an inability of them to migrate. And we also went on to show there's actually an inability of these cells to actually differentiate in this context as well. So we dove further, and this is just quantified here. We dove further at mechanism, and I don't have time to go through the entire mechanism of the data. I'm going to summarize it here. Unlike hair follicle cycling, epidermal injury and repair is a highly inflammatory process, and it turns out that regulatory T cells in this context use a different mechanism to actually facilitate hair follicle stem cell differentiation and function. And what Anu found was that it's actually, the Tregs regulate a specific module of inflammation at the CXCL5 IL-17 neutrophil axis, and that in the context of barrier injury, this axis is actually enhanced in the skin, and regulatory T cells suppress this axis, which actually allows the hair follicle stem cells to then differentiate and migrate and help repair the epidermis. But if you actually deplete regulatory T cells, this axis actually runs unregulated, and we don't currently know what about this axis actually potently suppresses the ability of hair follicle stem cells to differentiate and help repair the barrier. We're actually working on that. Suffice to say that this module is very important for Tregs to regulate in this context. Okay. So with that, I'd like to now just switch gears and talk a little bit about the translational aspect of our program and talk a little bit about some human work that we've done that I think has been very exciting over the last couple of years. So some of the technologies that we leverage in looking at human skin in I think a very relatively sophisticated and robust fashion is our multi-parameter flow cytometry. So this is a lot of our bedrock technology. We're able to take a very small piece of skin, 4 millimeter punch biopsy, which we have readily have access to, and we can look at the immune cells in these biopsies by looking at the cytokines that they produce using multi-parameter flow. We've also adapted CyTOF protocols and panels and established and sort of optimized them in the lab, where we have several different panels now where we can look at 40 parameters from one skin biopsy of normal skin to start to understand in a little bit more comprehensive way how the subsets of immune cells that are present in healthy and diseased skin. And then we've also been doing single-cell RNA sequencing, and we usually, what we do is we use our CyTOF panels to confirm interesting pathways that we identify using single-cell RNA sequencing. So these are just some of the techniques that we're leveraging to actually try to interrogate the function of lymphocytes that are tissue resident in human skin. And so using some of these technologies and our access to human tissue, we started to think about whether we could develop molecules that could augment Tregs in humans. And so, this would sort of, in my world, be the holy grail in terms of reestablishing immune homeostasis and immune balance in tissues. And I think that this will have implications for many of the diseases that we see clinically, autoimmune and chronic inflammatory conditions. And so, we focused this effort on trying on IL-2 biology. And so, as many of you know, and as I didn't mention earlier, but as many as you know, regulatory T cells were first defined by their high expression of CD25, which is the alpha chain of the high-affinity IL-2 receptor. And this was actually a pioneering paper, the pioneering paper on Tregs biology, which I hope Shimon will win a Nobel Prize for, published in JI in 1995, where Shimon Sakaguchi basically said these are a CD4 positive regulatory T cell population that express CD25. He defined them by their expression of the high affinity alpha chain of the IL-2 receptor. There's been a huge explosion of research in Treg and IL-2 biology since that time, basically summarizing to say that Tregs are dependent on IL-2 for their establishment, maintenance, and function. And so we focused on this IL-2 pathway and asked whether is there a way that we can preferentially augment Tregs using this pathway. And so before I talk about how we sort of did that, I want to talk about a little bit about IL-2 biology and IL-2 receptor biology because it's actually very complicated. I'm going to try to simplify it the best I can. But basically, IL-2, the receptor for IL-2 exists in two forms. There's the low affinity receptor, which is a dimeric receptor composed of the beta and gamma chains. That's expressed on a lot of immune cell types and other cell types as well. It's a broad expression. However, there's a high affinity receptor that is a trimeric receptor defined by the expression of CD25, which is the high affinity alpha chain. That, as I've mentioned, is primarily and constitutively expressed by regulatory T cells at high levels. So the dose of IL-2 actually really matters in this context. So high doses of IL-2 can primarily bind to the low affinity receptor expressed by a lot of immune cells. So there's broad expression, and it activates immune cells in a very broad and nonspecific way. And this is the approach that we've taken in dermatology to treat melanoma. Early on, high-dose IL-2 is used to actually treat melanoma by augmenting the immune activation arm, and with some positive results, obviously, but with a lot of toxicity, as we know. Newer approaches have capitalized on the fact of low-dose IL-2. As I mentioned earlier in my talk, that low-dose IL-2 actually has a predilection to bind to the high-affinity receptor, because when doses of IL-2 are limiting, it actually doesn't go to the low-affinity receptor, it goes to the high-affinity receptor. And the primary outcome of that binding is immune regulation. However, as you can see, you can't really push the dose, because if you start to increase the dose of IL-2 to try to get more Treg activation, you start binding to the low-affinity receptor and you get broader immune cell activation. So we began to think about whether a molecule could be engineered that might only bind to the high-affinity receptor and not bind to the low-affinity receptor, such that we could actually push the dose and get really selective Treg augmentation. And so to do that, we took an approach where we took IL-2 and we introduced point mutations in IL-2 and screened these point mutations based on the ability to only bind to the high-affinity receptor, the trimeric receptor, specifically not selecting mutants that don't bind to the beta chain. And I don't have time to go through all the screening process and how we isolate these molecules. Suffice to say that we were able to isolate a molecule that we believed had a high selectivity to only bind to the high-affinity IL-2 receptor, and we engineered that molecule to have a higher half-life. And so for the purposes of this talk, I'm going to call this compound mutein X, IL-2 mutein X. And so what does this compound do? Well, phosphostat 5 expression is shown here on the x-axis. Phosphostat 5 is a very sensitive marker of signaling through the IL-2 receptor involved in, and is actually a good marker for Treg activation. And as I've mentioned before, expression of the high-affinity alpha chain, the CD25, really defines Tregs. So these Tregs are in this blue box, high expression CD25, and the non-Tregs are the T effector cells, which are in the red box. And you can see that with IL-2, and this is just gated on human CD4 positive T cells, you can see that with IL-2, you do have dosages, the lower dose end here, you do get some activation of the Tregs, as evidenced by phosphostat 5 staining, but as you increase the dose of IL-2, you can see you get this potent activation of the non-Tregs, right, the T effector cells in the bottom, in the lower box. But with mutein X, we never saw that. And so as you push the dose of this mutein, IL-2 mutein, you're able to get selective Treg activation without the activation of the T effector cells. And so that's shown here in a dose response curve. You can see that this mutein actually selectively activates Tregs compared to all the other immune cell populations, with the exception of a very specific subset of NK cells, which I'm going to talk about in a second. Whereas IL-2, there is a window here where you do get selective activation of Tregs, but that window quickly closes and you get activation of all other immune cell types as you push the dose. So what we were able to generate is this protein, this IL-2 mutein, with greater than 3 log selectivity for Tregs over T effector cells and other immune cells. There is a residual fraction of NK cells that are activated by this molecule, but it's interesting that these NK cells are the CD56, CD56 bright CD25 high NK cells, which many have described as to be a regulatory population. So we actually might be getting double bang for the buck here. There's actually 2 regulatory populations that potentially are being generated by this, or augmented by this approach. And that we've now put this molecule into mice, into humanized mice in my lab, as well as non-human primates, and now it's in clinical trials, in early phase clinical trials. And so I firmly believe that this will be a mechanism whereby we will be able to restore immune balance in tissues of our patients. We can push the dose now and selectively augment regulatory T cells, capitalizing on the amazing work done around IL-2 biology. And I think that this will be efficacious actually in treating many of the patients that we see clinically by sort of restoring that immune balance in tissues. Only the future will tell whether this actually works, but I think it's a very exciting avenue of potential therapy. So to summarize the second half of my talk, unique subsets of Tregs reside in skin. Tregs play a role in multiple tissue intrinsic functions, and specifically they influence hair follicle stem cell biology. We're only really beginning to understand the cellular and molecular mechanisms of how these cells function in this tissue. It's going to be really interesting to see and discern the different subsets of Tregs, the different functions that they have. And one thing that I'm going to say here is that we do a lot of work in Tregs in mice, and we got to do a lot of Treg work in humans, and they're very different. So I actually believe that many of the things that we've uncovered in mice actually won't translate to humans based on the pathways that are active in human tissue Tregs compared to the pathways that are in mouse Tregs. And there's a lot of reasons why I think that might be, but we're seeing a lot of differences actually between the species in this specific cell population. And then, as I've mentioned, I think that a Treg augmentation therapy may be a powerful new modality to treat autoimmune and chronic inflammatory diseases and may impact the field. So with that, I'd like to thank members of my lab. This is our ugly sweater holiday party that they hate when I show, so I'm showing it. I want to do a quick shout out to Maggie Lowe, who's here at the meeting, who's going to talk about Tregs and hidradenitis suppurativa at the AbbVie Symposium. I'd like to thank my funding sources, my UCSF collaborators and outside of collaborators. I'd like to just give a shout out to the Dermatology Foundation, who's been instrumental in supporting my career. Just a quick plug as the Montagna Lecturer, there's also the Montagna Symposium that I think is going to be really exciting, and so I urge you all to contemplate attending that meeting. Thank you.
Thank you for that terrific talk. We'll take just 2 questions.
Mike, beautiful talk. Paul Neum from Seattle. So exciting, paradigm shifting, targeting the high affinity receptor. Just as a fun thought experiment, if this really worked and you could really goose Tregs, what do you think might be problems from having too many Tregs in certain organ systems?
Yeah, I think that's a great question, Paul. I think that, you know, the things that people immediately think of are cancer and infection. And the only thing, and those remain to be potentially legitimate concerns. However, I point to the data from Jerry Ritz, who's used low-dose IL-2 in his chronic GVHD patients now for 4 years, 5 years, a patient population that is prone to infection and prone to malignancy because they've had cytotoxic therapy and a previous malignancy, and he sees no increase in infection or malignancy in that patient population.
Great, thank you. We have one more, or I have one more duty. It is also my pleasure to give you this check on behalf of your lecture from the SID. Thank you so much. And I think that concludes the program. Thank you very much.